morning, dear colleagues. I would like to thank organizers for this awesome conference, and I present our study that will be, I think, interesting for many of us because a Crispr system is very well known. So, mitochondrial genome of plants is well known for its very large size and ability to acquire genomic content from the other organelles, such as plastids and nucleus. It has a structure with complicated rearrangements due to intra- and intermolecular recombination events. Despite of its large size, in comparison to other species, it contains the same number of genes. Part of sequences are imported from plastid, nuclear, viral sequences, and other foreign DNA. Almost half of sequences of mitochondrial genome has unknown origin and function. Another essential feature of plant mitochondrial genome is the ability to actively uptake foreign DNA. This phenomenon is well known and described in paper of my colleague a few years ago. You can see the name of paper on the slide. The CRISPR-Cas system of adaptive immunity is well known for bacteria and archaea, but we found the same CRISPR arrays in mitochondrial genome of plants. In fact, this phenomenon is poorly understood and described only in two papers. Francesca Magica, the pioneer of CRISPR-Cas system, 20 years ago described this CRISPR array in mitochondrial plasmid genome of Vicia faba. This paper has about 900 citations. And our paper 3 years ago was published in Vavilevsky journal about CRISPR arrays and Cas genes in genome of Arabidopsis thaliana. You can see on the figure a structure of this CRISPR array. The direct repeats are marked with bold text and red bars, and you can see that for two ecotypes, C24 and Landsberg erecta is the same, but for Columbia ecotype is disrupted by mutations and insertions. We studied more than 200 of mitochondrial genome of plants, more than higher plants. And in 130 of them, we found a CRISPR arrays. This species includes agriculture, wild species, model species, and even liverworts. Interesting that this number of CRISPR arrays doesn't correlate with the mitochondrial genome size. The diagram will be on the next slide. Some of mitochondrial genomes contain dozens of such CRISPR arrays. For example, Cucumis sativus contains more than 50 of them. Distribution of length and number of arrays. One can ask, maybe this CRISPR array is just a repeats and you have false positives, but we studied the conservation score of these sequences, and conservation for direct repeats differs a conservation score or entropy for the spacers. You can see they are very different. Additionally, we analyzed the spacers that extracted from these CRISPR arrays, and many of them have homology with mitochondrial viruses sequences. Unfortunately, these mitoviruses are very poor understood, and there are only few papers about them. You can see the three of these papers on the slide. that led us to suppose that the spacers can do the same function as in the CRISPR-Cas or bacterial immune system. We also analyzed the overlapping of CRISPR loci with the existing annotations in mitochondrial genomes. Most of them overlaps with the coding sequences and introns. The CRISPR direct repeats have a secondary structure called hairpin. We built this secondary structure for our sequences of the direct repeats, and you can see that is pretty the same, part of them pretty the same, such for bacterial ones. And on the next slide, you can see the two largest clusters of these repeats. It forms a hairpin, a pretty hairpin that's like a bacterial one, and its repeats form a clusters among species. The next largest cluster. 3 CRISPR arrays have copies in mitochondrial genome of Triticum aestivum. Interesting, that is contained into a repeated region, and we suppose that it's putative remnant of ancient linear plasmid insertion. This phenomenon is known for mitochondrial genome plasmids and described in a paper of Daniel Sloan and colleagues. You can see on the slide. So we go to the conclusions. My talk is very brief. We discovered putative CRISPR arrays that has homology in different species and RNA secondary structure. Many of them form the structure, form clusters within species and form the hairpins. We can suggest that this system has the same function as for bacterial ones. The number of these CRISPR arrays does not relate with the length of mitochondrial genome size. Some species, cereal species, used to have linear plasmids with terminal inverted repeats possessing the CRISPR array, like ones which are described for maize plasmid, three plasmids, and one of them linear 2 and 3 kilobars size. So this talk is the first step of study this cryptic phenomenon, but we also have found the CRISPR arrays in Synaptus elegans, higher plants nuclear genome, and even mitochondrial plasmids of Aryza sativa, the maize, and other plasmids. Thank you, and I would like to answer your questions.
Thank you for your presentation. Please, questions? Okay.
Thank you very much. And I wanted to ask about those mitoviruses. You said that there's little information, but maybe you know those viruses are like domesticated, or there are viruses which can invade, I mean, real bacteria or some prokaryotes.
Thank you for the question. This virus is poorly studied. They are discovered in silico only. These papers describe only sequences and doesn't describe any function or hosts among kingdoms or species or.
Maybe it can be predicted at least the strategy of replication, I mean, recombination of the chromosome, ability to latent phase or something like this? RNA viruses, DNA viruses?
I don't understand your question. If maybe in coffee break we discuss this further.
Oh, thank you so much.
Next question, please.
Thank you very much for your talk. I would like to ask a question. So you have found possible CRISPR arrays in mitochondria of plants. So have you tried to screen the open reading frames in near vicinity of those CRISPR arrays for Cas1, Cas2, or other very conserved proteins of CRISPR systems?
Yes, sure. But these genes are related to bacterial ones, and the introns and different expression system in eukaryotes not allows us to use the software that was developed for bacterial.
But if you consider the theory of bacterial origin of mitochondria, maybe we can try at least as an attempt to use the bacterial software to.
Yes, we located some genes. You can see in the paper.
Okay, okay. I'll check the paper out. Thank you very much.
Okay, one more question.
Thank you for report. So question, as of multichromosomal and complex structure of mitochondrial genomes of plants, did you note any differences in gene origin and CRISPR activity between chromosomes?
As I know, there are only circular structure of most of mitochondrial genomes. One chromosome, circular.
There are noted multichromosomal cases in plants in literature. Yeah. Maybe you can say them plasmids. Some of them may be circular and another linear, but they may contain genes and may have a different origin. So did you noted any differences in origin of these fragments?
Thank you. We doesn't consider their origin at this step, but we found a CRISPR arrays in mitochondrial plasmid already. There was a report last year on plant gene. And my colleague, Igor Gorbenko, that was co-author of this study, reported about this topic.
Okay. More questions? Please.
Thank you for your talk. My question is the following. According to the data you have already, do you have any hypothesis of what is the function of CRISPR repeats you found in mitochondria? Is it close to bacterial, or probably it's different, or probably it does not function there?
Mm-hmm. Thank you. The most obvious function is the immune system, and we checked their homology with putative viruses, and also checked with other viruses sequences. But how to discover the immunity effect? It's not a simple question, but we will continue this study to use the transcriptomic data to found the gene that can support the system, but it's a very complex study because I have any experience with expression studies and transcriptome.
Okay, and I have one more question. So you show that you calculated the secondary structure of these elements, and are you sure that that RNA from these loci are expressed first, and they are properly post-processed to obtain such short RNAs? Because if it's just in the genome and it's not processed to RNAs, there is no sense to talk about the secondary structure.
Thanks. As I know, almost all content of sequence of mitochondrial genome is expressed. That's why we doesn't check this fact, but it's obviously need to do to check the transcripts of following clusters to ensure that this RNA forms a real hairpin, of course.
Okay. Thank you.
Colleague, I ensure you to talk in a coffee break.